All right, group number 3 is amino acids and proteins. So we did lipids, we did carbohydrates. Now we are on amino acids and proteins. Now, proteins are the things in the body that do this stuff. Okay, so we're going to talk about what those are like monomer wise and how they build together to make bigger ones. So I just have some examples of classes of proteins, what they do, and some examples of specific ones. So I've kind of underlined ones that we talk about a lot more. Enzymes, for example, we especially talk about enzymes when we're talking about speeding up or slowing down reactions. They catalyze, right, speed up, and control biochemical reactions. One example of an enzyme that is a protein is DNA polymerase. That's how we replicate our DNA, and you have to be able to replicate your DNA to create new cells. We've already talked a little bit about hormones as well, and hormones were types of steroids under the lipid family, but there's also hormones that are proteins. They regulate metabolic processes. That's like the function of hormones. So the lipids also do that. They regulate metabolic processes, but protein hormones do the same thing. An example of a protein hormone is insulin, right? Insulin is what regulates our glucose metabolism, breaking down those glucose molecules into energy. So of course, we need to regulate that. And if you have insulin issues, then you have diabetes. So how do we make these proteins? Okay, so we're going to start small again, right? Amino acids, those are the monomers, or we call them the building blocks of proteins. You stick a bunch of amino acids together and you end up with a protein. So an amino acid looks like this. It has some very specific parts to it. Okay, it has a carbon in the middle. We call this the alpha carbon. On one side, it has a carboxylic acid, and on the other side, it has an amine. Okay, so C double bond OH and an NH2. And the fourth thing bonded, we'll just call an R group because it's different for different amino acids. This is what changes between amino acids. Everything else is the same. There are 20 amino acids, and this alpha carbon is always chiral because this group is different. All except for glycine, because glycine's R group is just hydrogen, so it's not technically chiral, but the rest of them are chiral. The 20 amino acids all have a 3-letter abbreviation. You are not going to have to memorize these, but I will show you them, and sometimes we'll use them when we're talking about amino acids. But what differentiates all the amino acids is this R group. Okay, so for example, I've got alanine, serine, and lysine here. These are all different amino acids. Now, I kind of have the R groups drawn upside down. You'll notice this is my carbon. I have an amine over here. I have a carboxylic acid over here. I have my hydrogen up, and then my R group down. Alanine is this amino acid, and it has a methyl group, is its R group. Nonpolar group, right? Methyl is nonpolar. This is what changes the properties of all of our amino acids. So serine, on the other hand, looks like this. Its R group is this guy. Okay, notice it's got a carboxylic acid on the end. Okay, so this is going to be a polar and acidic R group because it has a carboxylic acid on the end. Causes it to act different ways. Lysine, on the other hand, over here has this group, okay, which has an NH2 on the end, making it basic. So we can also have basic R groups, and that causes that amino acid to attract to different things and act certain ways. So this is how proteins really get stuff done, is by chemical reactions between acidic or basic or polar groups, things like that. Okay, so the 20 amino acids are right here, and no, you do not have to have them memorized, but I really like this picture because it differentiates all of the 20 and the differences between them. So alanine, for example, we were just doing alanine. The way that these ones are represented, the chiral carbon is right here. I have a double bond OH. This is the chiral carbon. This is the chiral carbon, sorry, with my carboxylic acid over here. My amine group in this one I have going down, and I think most of these are like that. And then the R group is off the side. This one, the R group is CH3. Glycine, I mentioned, only has an H as an R group, so it actually looks like this. It's got the NH2 up there. But the rest of these all show the NH2 going down, and then the R group on the left. So isoleucine, this is its R group. Leucine, this is its R group, so on and so forth. So we start getting some that are more polar, some that are aromatic, right? These have some benzene rings on them. Some that are acidic, those are the ones I kind of starred because we talk about acids a lot. Aspartic acid and glutamic acid, they have carboxylic acids there in their R groups. You can also have basic ones. Okay, so basic ones have a nitrogen there in their R groups, nitrogen there in their R groups. There's a whole bunch of different kinds, right? The other thing you can notice about these is some of them have a solid ring and some of them have a dotted ring. And that's because the dotted ones are essential ones, meaning you have to consume them in order to survive. So we have to ingest those in some way or another. Okay, so now we're going to start putting these amino acids together. How do we do that? We form what is called a peptide bond. Okay, so formed between the carboxylic acid of one amino acid and the amine of the other. So I have a amino acid right here with an R group, whatever it may be. This is my chiral carbon. I have my carboxylic acid end over here. And then this is my second amino acid. I've got my amine group here. Now, when you have a carboxylic acid bonding with an amine, we make an amide or an amide bond. Okay, we take out a water and now we have a C double bonded to an O to an N to the rest of the molecule. This is something we did in organic. So this bond right here is formed when the water is pulled out. So water is one of my products. And this is also called a peptide bond. So what type of bond is it? It's an amide linkage or an amide bond. But we call this a peptide bond. So we start sticking amino acids together. And this is the peptide bond. So then when we stick all of these together, you'll have a big long chain of amino acids. One end would have an extra NH2 on it. The other end has a carboxylic acid on it. We call this end the N-terminal end, and this end the C-terminal end. And so that's how we differentiate between the two ends of a peptide chain. So a polypeptide means a bunch of peptide bonds, right? A bunch of amino acids bonded together. So after you have two, this is a dipeptide. Then anything bigger than that is considered a polypeptide. So 3 to 50 is a polypeptide. Once we get past 50 amino acids bonded together, then we start referring to them as proteins. So sometimes we call proteins and polypeptides interchangeable, but it's really that proteins are like the big ones. Okay. And how do we get these proteins? Well, we had DNA that codes for the amino acids to create a protein. So everything kind of plays together, right? And we'll talk more about DNA in a little bit, but that is how we code for creating a protein. Now, protein structure is really important to give proteins their purpose, what they do, how they can react. So different structures do different things. And there are multiple levels of structure for each protein. So all proteins have a primary structure, and a primary structure just is the sequence of amino acids. So if I write out the sequence of amino acids, that's the primary structure of the protein. That's what we get from the DNA molecule. Now, when you build a protein, however, some of those R groups on your amino acids start to interact. Some of them attract each other. And so we get what are called secondary structures. And there's 3 types of secondary structures that are most common. There's an alpha helix. You'll notice it kind of looks like a corkscrew, like a helices. And that's because we get these hydrogen bonds between polar groups. So we get a corkscrew shape. It happens because of hydrogen bonds every 4th amino acid or so. And then the rest of the R groups are going to kind of face outward. So we get hydrogen bonds, we get a little corkscrew. That's called an alpha helix. It happens sometimes in molecules. Also, beta-pleated sheets. This is a different type of secondary structure. It's when you get rows of amino acids that again are held together with hydrogen bonds. Okay, hydrogen bonds do a lot of stuff. So they kind of start packing together, and that's when we get a beta-pleated sheet. This is a very rigid structure. There's also triple helices. I don't have a picture of that over here. Not as common, but it's when you have 3 peptide chains kind of woven together like a braid, and you get hydrogen bonds. So these are 3 types of secondary structures. So the amino acid sequence is made, it forms some secondary structures, and then those secondary structures interact to form the tertiary structure. This is the overall 3D shape of a protein. What does the protein look like? And there's generally 2 types of tertiary structures. There is a globular type protein or a fibrous type protein. Kind of what they sound like. Globular is like a glob, right? It's a compact, it's a round shape. So some proteins kind of glob up like that. When they do, their non-polar R groups kind of point out. The polar ones go in, they attract other way around. Non-polar points in because there's water on the outside. The polar ones point out, making it soluble. Molecules that look like this that are proteins are hemoglobin, insulin. They need to be able to get from point A to point B. They're like little globs of protein. And fibrous is when they are long and thin. So you can like think of fibers. This is keratin, your hair, skin, and nails. That's where keratin is. Of course, your hair is made from long, thin, fibrous proteins. It's not a bunch of globular proteins. Now, how do you get different hair then? When you have things called like cross-linking between those proteins, you get curly hair. Over here is just kind of a picture of main tertiary structures. So the overall, right, big structure is a tertiary structure. Maybe I have some secondary structures on the inside, some alpha helices, some beta sheets created by this hydrogen bonds. I can have things called disulfide linkages. We can have all kinds of stuff going on. But that's like where you get kind of like the cross-linking. But this would be like the tertiary structure, the big overall three-dimensional shape of a protein. Okay. So this is a little picture of an overview, right? Our primary protein structures are our amino acid chain. Secondary protein structure are things like pleated sheets or alpha helices. Then they start attracting each other, and then I get my tertiary structure, which is my 3D, what does the protein look like? And then sometimes proteins bond or attract with other proteins to create a quaternary protein structure. Insulin is an example of this. It's when you have multiple proteins coming together to be one big molecule that does something. Insulin is made up of a couple types of proteins that come together and they attract each other or bond, and then they can do whatever it is they're supposed to do. This is an example of kind of a protein chain that's got some other stuff going on. This is a lysozyme. A lysozyme kills stuff. It's like an antimicrobial. And it's got some, like we got a disulfide bond here, disulfide bond here. This kind of looks a little globular in nature. It's just kind of an example of what you would see with those proteins. Insulin itself, I like talking about insulin because obviously it's extremely important in the body. It was sequenced in 1955, which was a long time ago. And then by the 80s, they could use bacteria to produce the same sequence as human insulin. And that's how we get our stuff. We would use plasmid DNA and bacterial DNA and stick it in a bacteria. And then it would produce all of this, all these new molecules, and we could take out the human insulin and then we can use it. So the sequencing of insulin was extremely important. Okay. I think that label was here for insulin. Yeah, I don't know if it's quaternary or not. Hang on. Yes, I just double checked myself that insulin does have a quaternary protein structure, multiple units come together.